Thank you very much for the opportunity to come along and speak to you, at least virtually. I'm sorry not to be there physically. In fact, I was meant to be talking to you about almost exactly 1 month ago, but the meeting was deferred. At that time, I think you were hoping to resolve your leadership discussions.
Yeah.
When it was suggested it be deferred, I did wonder whether at this time you would yet know the answer. And I suppose you got closer to that than you were then, but perhaps still not entirely certain. Also at that time, I thought the likelihood of a vaccine occurring before I gave this talk was infinitesimally slow, unlikely. But yet it seems science works faster than politics, and we have vaccines, and yet not quite the resolution that I think you hoped for a month ago. It's really exciting to talk about the opportunities, I think, that are ahead of us in terms of the reinvention of randomized trials. And I use the title Evolve or Die, the urgent need to streamline randomized trials, to really talk about how we might do randomized trials if they were invented today in the 21st century. So when we think about the underlying themes of trials in terms of what are the things that are considered to matter, well, obviously protecting the patients in the trials, their safety, their rights, their well-being, but also importantly, protecting the safety and well-being of people who are treated as a consequence of clinical trials and other studies evaluating treatment. I think the problem that underlies a lot of the regulation and guidelines that have grown up over the last 20 or 30 years is that in the attempt to achieve these aims, I don't think that effective strategies have been put in place that are both proportionate and efficient or effective. And I don't think I'm alone in that either. The European Commission, which in 2001 launched what was called the European Union Clinical Trial Directive. A directive is different from a regulation. It's a set of principles or guidelines that need to be implemented into law in every European country, but can be implemented in different ways. Their conclusion 8 years later was that that directive, instead of improving the quality of the trials that were done, had increased cost without added value, increased the demands on scientific staff due to the inefficiencies of what they had put in place, that it had been implemented differently in different countries to the detriment of the safety of clinical trial participants, and that inconsistent application that was occurring both between and within countries was stopping patients from having access to new treatments. All the opposite of what had been intended. And they concluded in their review that as a consequence, performing clinical trials had become considerably more difficult and costly to the detriment of the European population. So when one thinks about what has been the adverse impact of increased regulation and related bureaucracy on randomized trials, there's this first one of increasing obstacles and delays and costs for trials. And I think at least in part, that has encouraged many to think about what might be alternatives to doing randomization, and in particular, whether it was possible to replace randomized trials by using observational studies. The emergence of large databases, very fast computers, and an increasing array of statistical approaches, I think encouraged that use. And of course, it avoided all of the complexity that had been put in place for randomized trials. So the United Kingdom Academy of Medical Sciences, I suppose the equivalent of the National Academy of Medical Sciences in the US, wrote a report in 2015 proposing non-randomized real-world evidence as an alternative for generating evidence about randomized trials for these very reasons. And in 2017 and 2018, the National Academy of Sciences ran a series of meetings and put out 3 reports, really along the same lines. That could we find an alternative to randomized trials? Well, as epidemiologists involved in research will be well aware, observational studies are incredibly valuable at identifying causes of disease. We know that smoking causes lung cancer, many other cancers, cardiovascular disease, respiratory disease, as a result not of randomized trials, but observational studies. We know in this example here that statin therapy causes the rare but serious complication of myopathy. Beneficial effects, large beneficial effects, penicillin on pneumonia, or defibrillation that have large effects on rare outcomes, where survival is unlikely, you give a treatment, survival occurs very often, do not require randomization. But where we're looking for moderate effects on common outcomes, I think that the underlying problems of confounding and bias in observational studies is often underestimated. And although very large scale databases do provide the opportunity to obtain very precise estimates, that is, estimates with small random errors around the point estimate, that doesn't really necessarily provide good evidence that those associations are causal. Here's one example. There have, over the last couple of decades, been a very large number of randomized trials of cholesterol lowering with statin therapy, several coordinated out of Harvard, the CARE study in myocardial infarction patients, most recently the Jupiter study coordinated by Paul Ridker in primary prevention. There have been over 100,000 patients in those trials, 5 years of treatment. And when one looks in the combination of all of those data, very large numbers of individuals have developed cancer. We have really reliable evidence from these trials that during 5 years of treatment and follow-up, there is no effect whatsoever on cancer, 5,060 versus 5,064, and no effect on any particular site of cancer. And indeed, many of these trials have then followed patients up long-term through linkage to health record systems and shown that 5 years of treatment has no effect on cancer. And yet in 2012, in the New England Journal, there was a claim made based on what in many ways might be seen as the perfect observational study from a whole population in Denmark of very detailed health record system, suggested a 15% reduction in cancer associated with statin use. However, in the same paper, statin use was associated with an increased risk of cardiovascular disease. Now, we know that statins lower the risk of cardiovascular disease. We know that this result is wrong. And therefore, when we have a result like this, it's not that difficult to adjust it away because we know it's incorrect. And that's what was done in this paper. But that does not guarantee that the adjustments that are done to get rid of the confounding that is producing this aberrant finding for cardiovascular disease, that those adjustments are necessarily adjustments that will deal with the confounding or biases that are suggesting an effect on cancer, which, as I showed on the previous slide, is not supported by the randomized trial evidence. And I should say that although people often refer to observational studies as having not just large scale, but long exposure, the average exposure in these studies was only a couple of years. So as a number of people, including Dr. Sherman from the FDA said, essentially the availability of data, the availability of analytic tools does not necessarily mean that using these observational data will provide reliable evidence, and really stated a concern about the problem of generating incorrect or unreliable conclusions from such data. So although there is clearly a place for observational studies in understanding causation, both good and bad effects when they are large on rare outcomes, I think for a lot of the treatments that we have, moderate beneficial or adverse effects on common health outcomes require randomized trials. And so rather than trying to get around the regulations on randomized trials by doing some other kinds of studies, I would suggest the approach should be to get rid of the obstacles to doing randomized trials in the first place, and to do trials that also include a wide range of individuals, which is often a criticism of the trials that are done currently, in order to yield greater generalizability. Not only has this increased regulation and bureaucracy resulted, I think, in a mistaken conclusion of a direction of travel, but I think it's had a terribly detrimental effect on the development of new treatments and also the evaluation in non-industry trials of current therapy, finding out whether what we do now is really a good idea or not. Here's a analysis done by Clive Meanwell, the medicines company, looking at what has happened over the last 15 years in terms of the pipeline of treatments that have been developed by industry. So on the top lines under revenue, if you work in the industry, you think that's very good. It's gone up from 2000 to 2015, from $34 billion to $84 billion a year with the top 10 drugs. So it's more than doubled. But if we actually look at the numbers of people that those treatments are benefiting, it's gone down from just over 400 million to about 50 million, a 7.5-fold decrease. So what we're getting is a move away from population targeted treatments towards niche targeted treatments. Very, very good treatments, but for a smaller and smaller group of individuals, and the treatments costing more and more for those individuals. I'm not arguing against having these niche treatments, but what I'd like to see is the benefits of both. I think also we've seen in the last 15 years the regulation and the anxiety of being seen to comply with the regulation, with audit by regulators. The funders of trials, often industry, being very much less likely to work with academia in a collaboration to create the most reliable evidence for the most patients. And indeed, what we've seen instead is the creation of a new industry, which in 1990 and 1995, when the International Conference on Harmonization was created, and then they put out their ICH Good Clinical Practice Guidelines in 1995, an industry which really didn't exist at that time has been created, it seems. I take the criticism that it's difficult to be sure of causation, but it seems that there is an association with the creation of a $40 billion a year industry that is dealing with the cost and complexity of the trials by making them more costly and more complex. And the focus on complying with rules, as with the example shown with the review of the EU directive on clinical trials, is that rather than looking at innovation and design and conduct of trials, the focus has been on compliance with a tick box mentality. And the European Society of Cardiology in 2013 did a report on this and concluded that the regulatory barriers to properly conduct clinical research in Europe currently abound, and it can be mind-bogglingly difficult to conduct clinical trials. All of these things making it less likely that we will evaluate both current and new treatments. And I think also pushing academia away from the evaluation of treatment in reliable trials. So I think one of the biggest drivers of this has been the ICH GCP guidelines. ICH is a creation of 3 regulators, the FDA, the European regulators, and the Japanese regulators, and industry. It was created in order to have a single set of guidelines for the registration of new drugs. They wanted one set of rules, didn't matter how good or bad the rules were, only that they were the same in all of the major markets. It's not about science, it's about marketing and getting registration and selling the drugs. So they're not based on the key scientific principles that underlie randomized trials and the matters that are critical to the generation of reliable results. Here are some examples. The guidance on monitoring of collaborating sites. So it says the purposes of trial monitoring are to verify that reported trial data are accurate, complete, and verifiable from source documents. Well, the fact is, in a randomized controlled trial, it isn't actually necessarily the case that high quality data will mean that you get a reliable result. If the trial was too small, for example, you could have perfect data and not get a reliable result. On the other hand, the corollary is also not true. You can get a reliable result even if the data are not perfect. And this is because of the beauty of the randomized controlled trial. You have randomization, which means you're comparing like with like between people who get treatment and a control group. So interestingly, missing data have little impact if this is unbiased with respect to the allocated group, missing data about health outcomes. Here's a made up example, but let's say we were doing a large scale trial in cardiovascular disease, 10,000 given the new treatment, 10,000 control, and the treatment reduced the risk of, let's say, mortality by 20%, from 1,000 deaths, 10%, down to 800 deaths, 8%, so 20% reduction. Well, what happens if we missed some of these real events? Well, they're real, so the treatment affects them, so they would be unevenly distributed. If we miss 10% of them, it would be 720 versus 900. Still a 20% reduction, still highly significant. If we miss 20% of them, the same would be true. So the beauty of the randomization and the control is that it's incredibly robust to, in this case, missing events. And yet enormous amounts of effort go into trials because of this focus on data rather than reliability of findings. It's also the case that false positives have a remarkably small effect. So again, a massive effort is gone into in clinical trials when a patient reports an event. If it's one of the main outcomes of the study investigators will often go to great efforts to get hold of the clinical records. They will fly in a set of experts in that particular outcome who will sit around and determine whether or not the patient was right or their doctor was right. And that actually costs a lot, but typically adds little. So here, if we add false events, so they're false, so they're not affected by treatment, they're evenly distributed, add 10%, add 20%, and as you can see, we don't make a jot of difference to the evaluation of the treatment. Now, these are made up numbers, but what about some real numbers? So here is a trial we did, the heart protection study of 20,000 patients, looking at the effect on coronary events, strokes, and revascularizations, a 23% reduction in risk. Overall, the diamond at the bottom. All of these events, we went and got information about them from their doctors. Several people then went through all of that information to adjudicate them. And this is the result after all of that. So before, after, before, after. You can see very little change through all of that adjudication, which was a large and costly effort and repeated in many trials. it really fails to take advantage of the power of the randomized controlled comparison. Again, in randomized trials, the guidelines have a lot of focus on safety monitoring, recording all the adverse events, not just the serious ones, even when it's known what the treatment does to less serious outcomes. Narratives on all of these are often recorded and reviewed with little benefit. Demands for unblinded results for adverse events during ongoing trials. And we've seen that most recently in the pandemic with the EMA, the European regulator, and the MHRA, the UK regulator, demanding on the basis of observational studies of hydroxychloroquine, which, by the way, turned out to be fraudulent, but based on those studies, they demanded that the randomized trial being done by WHO, the Solidarity trial, and the one being done in the UK, the Recovery trial in parallel, be stopped and unblinded because the observational studies had indicated a big hazard, which was not confirmed by the randomized trials, which went on to provide compelling evidence. So one of the big problems is I think the regulators themselves don't really understand the randomized trials. And lots of annual reports in order that those safety signals are identified. And I remember having a conversation with the UK regulator, the MHRA, and I said, what do you do with all these safety reports? And they said nothing, because we just get swamped by them. We can't see the wood for the trees. And here's another example about the pointlessness of a lot of this. So a trial we did of niacin in some 30,000 patients, just looking at all of the adverse events that were recorded as part of that trial, 3 completely unexpected findings came up. One was that niacin caused new onset diabetes, it caused hospitalization with infections, and it caused hospitalizations with bleeding. And they were quite large effects, 30% increases. Now, niacin had been used for 50 years, particularly in the US. It was actually about $1 billion a year sales in the US prior to these results. Many observational studies had been done of niacin. They had never detected these moderate effects on relatively common outcomes. They were certainly not detected during the trial by the regulatory requirements for the reporting of so-called serious unexpected, suspected unexpected serious adverse reactions, SUSARs. Another recommendation is that it's necessary to go to sites both during and after the trial to identify problems with the trials, and only in exceptional circumstances use central monitoring of the process. I think this is exactly the opposite of what the evidence demonstrates. There's very good evidence actually that going to sites is not effective at detecting problems, and that central statistical monitoring is effective at detecting anomalies. And yet, the site monitoring has been estimated to be about 1/3 of the cost of large-scale randomized trials done by industry. It involves going and checking people's CVs to see whether they're qualified to do the trial. Well, if they can't make up a fraudulent CV, they're really not that good, are they? That they've done their good clinical practice training. All of these things are tick box, reviewing consent forms, but not actually reviewing the process of informed consent to actually understand whether the person has been properly informed. And we've just seen longer and longer information leaflets that are probably reducing further patients' understanding of what they're signing up to. Lots of data verification of things that are not important to the trial, documentation, pill counts, etc. Many of these things just irrelevant to producing reliable evidence from randomized trials. By contrast, if we thought about how we would do randomized trials now, we wouldn't as with this talk today, we wouldn't get on a plane and fly to Boston to give the talk and then fly straight back. We would look at the data, or we would look at our screens to give the talk. We would use the systems that exist now to improve the quality of the studies. And on that plane, there will be systems that are listening to the sound of the jet engine. And if they hear a change in that sound, then that's when the engineers will look to see, is there a problem? They're not going to wait and do it every 6 months. They're going to use the data to identify problems. And here's an example where one can look to see for all of the sites across a study for analyses of kind of key indicators of how the trial is being run to pick out those sites where things are different from the others, the red blobs here, where the blob is the size of the blob is indicating the size of that center. So you can see there are a few where you would like to understand why are they different. They might be different for good reasons or bad reasons, but they are different, and that can help to direct the kinds of visits that one does and what one looks for. So the whole idea should be about identifying problems early and helping staff not to make errors, rather than on finding the problems after the event when it's too late. And I think that's, again, where one comes to how one would approach the way in which we do clinical trials now, where we don't have paper record systems. We do everything online. We turn our protocol, or we should, into essentially an electronic system, the protocol writ electronic, that actually manages the whole process to aid compliance with the study protocol, not just by participants, but by the staff, to help ensure complete and comprehensive data are collected. If particular answers are given, to ask additional questions, to have codes to ensure that there aren't anomalies between the data within a visit or between visits. And then, of course, that data can be looked at in real time to monitor and ensure compliance with the protocol and high quality. But instead, what we've seen is these principles which were set up with a marketing intention, not based on the science, by the regulators and pharmaceutical companies for registration trials of new drugs being applied to all trials. So they are now a requirement, ICH GCP, of the European Union regulation, which followed the directive, but also of funders, the Gates Foundation, NIH, Wellcome Trust, require compliance with guidelines that were set up for completely different reasons and not based on the scientific principles. And they're not even working well for the registration trials of new drugs. As I say, unsustainable costs, wasteful practices, and indeed, poor quality by not taking advantage of the systems we have available to us now. Here's an example. The FOURIER trial of PCSK9 inhibitor therapy, the antibody. I mean, PCSK9 inhibitor therapy is an extraordinary achievement from genetic data to late stage clinical trials within not much more than a decade is extraordinary. And yet it's been a failure of treatment development. This single trial cost more than $1 billion. There were parallel trials going on. The companies were in a race to get to market first. The cost of doing the trials and keeping them going was so high that although they recruited 28,000 patients, they only kept treatment going for 2 years, despite our knowledge that lowering cholesterol takes time for its benefits on cardiovascular events to emerge. As a consequence, they didn't allow the benefits to emerge. They underestimated the reduction in risk by only allowing a couple of years of treatment, and failed to demonstrate an effect on mortality, providing an opportunity for payers not to use this treatment. It should never have cost that much. It should never have gone on for so short a time. we're really, with these guidelines, we are damaging enormously the improvement, the possibility of improvement in healthcare. ICH has now finally recognized that there is a problem and are in the middle of a detailed revision of the guidelines. But again, that's being done by industry and by the regulators. It does not involve at its core the patients or academic trialists, or indeed anyone who's actually conducted successful clinical trials. So I think that there is a solution to the problems that have been created, but that solution is to make randomized trials better and easier to do so that we get reliable evidence, and that we then combine that with observational data in a way that allows us to extrapolate from the randomized trials to different populations. So in conclusion, what are the opportunities? What could happen if we imagined that the randomized trial was invented today? Well, I think what we would do to start off with is explain the underlying scientific principles, the robustness of randomized controlled comparisons, of the comparison of like with like in terms of the patients, the comparison of like with like in terms of the way in which the health outcomes are ascertained, that allows data that are not perfect to provide really reliable evidence about the safety and efficacy of treatment. And I think that we do now need to do this in partnership between regulators, academic and industry researchers, and importantly, of course, patients. We need to look at ways in which to enhance recruitment. It may be that one of the few positives that come out of the last year is that the public will see science, medical science in particular, as being a really positive thing, and that the evaluation of treatment in randomized trials and of vaccines in randomized trials has been literally a lifesaver. So it may well be that we could build on that and encourage patients to be more willing to join randomized trials, that we can use the health record systems that are available to us, not to try to evaluate the effects of treatment in those observational data, but to recruit patients into randomized trials more efficiently, to be able to include a much broader and generalizable set of patients into those trials, to improve the quality of the trials by using electronic trial management systems, as I outlined, to help people who are involved in the trials to do them better and more enjoyably, and to use central monitoring approaches to again improve both patient safety and the performance of the study. And finally, the health record systems provide us with an opportunity to find out what happens in the longer term. So by linking into the health record systems, we can not only improve the quality of the data that we get about patients during the trial in terms of all the different health outcomes and the direction of the effects, good and bad, on those health outcomes, but also what happens in the longer term after people have been treated within a randomized trial. And finally, I think there are opportunities to evaluate the effects of treatment on patients through direct approaches to patients, through, say, web questionnaires or through devices that they might wear that would allow us to get even more information about the effects of treatment on a range of different things that matter to patients. This is something that we described quite recently in a New England Journal commentary piece. Thank you very much for your attention.